Thank you very much. First of all, good morning, everyone. Uh, Let me thank Ruchin and Tom and the BioEM Talk. Uh, these are the BioEM Talk organizer for giving me the opportunity to present my work here on this platform. And I'm thrilled to be here. And I'll be talking about the ants, mosquito, and electron microscopy, and half an hour of challenges and triumphs. So, uh, So let me set the stage. I work at the Electron Microscopy Resource Center at the Rockefeller University, uh, in New York. It's a very big operation and run by two people, uh, Anurag Sharma, that's me, and the director of the core, Hilda Malia Pasoli, a fearless one. And we are specialized in making processing wild world of the biological samples, sample preparation and imaging them and discussing with the users the outcome. So yeah, so let me introduce our stars here, the ants, which I'm going to talk about. So this is the Ocirobiroi. These are the clonal radar ants. And Daniel Cronauer's lab is working on these ants, various aspects of these ants, the life cycle and the different stages of ants. And these are the researchers from the lab. I'm going to show the work today. And this is the another lab, Leslie Washel's lab, which the lab works on the Aedes aegypti mosquito, various life cycle, and the other various aspects of the mosquito's life. And Leah is the postdoc from the Waschel lab, which collaborate with us to do scanning electron microscopy and TEM to know the answer of the research she is pursuing. So just to give you a brief introduction, that the clonal radar ants, actually they are the social insects, and their life stages goes from the egg to larvae to pupae and the adults. And imagine that they have a big colony and only there are females, there are no males in the colony, and they have no eyes. So they have to, rely on their, well-developed sensory system or sensilla actually to receive signals from surrounding and to or one another. So how do they perceive signals to different sensilla and transmit to the brain is one of the major questions Daniel Cronauer lab is taking in account, and he's also interested in like studying various factors governing the maintenance, the growth of the colony. Okay, and on other hand, Leslie Boschel, she works on female mosquito, Aedes aegypti, and these these female Aedes aegypti they are cause of spreading many diseases like yellow fever, dengue, Zika viruses, and many more diseases. And they hold the crown for like the deadliest female on the planet. Um, they fe- feed on the human blood just to nourish the eggs. So Leslie's lab is interested in decoding their feeding behavior and the human blood feeding behavior and how they interact with the human. And besides that, I would like to point out that these Egypti females, they mate only once in a lifetime. And Leslie's lab is also focusing on decoding their mating behavior and the brain signaling. Brain signaling. Okay, so a few years back, a researcher named Orly Snare from Cronauer Lab, she was interested in she discovered actually a process happens in during the pupal stage of the clonal radar ant. So the pupa of clonidal ant, they secrete a nutritional rich molten fluid, um, and this is a very rich in nutrient, and adult drink it and also feed this fluid to the larvae for their growth. Uh, so it is very rich, and this is like drinking is indispensable for maintenance and the growth of the colony. So Orley was interested in seeing the morphological characteristic of these pupae. So what we did, we fixed these we fixed these pupae by conventional electron microscopy method uh, for scanning electron microscopy, and image them under scanning electron microscope. And you can see that this is the nicest structure, false colored pupae. And what we found, like in the terminal part, so this is the terminal part of this is the zoom in of the terminal part of the pupae. And we found out that there is like anus is one hole present in the terminal part. But this fluid, what the pupa secretes, it secretes, they have a specific opening for the secretion. So what we concluded that this fluid is not directly coming from the digestive tract. It has some other opening or some other uh, way to come out. So this is what our SEM images could confirm. And so this study was so fascinated that scientific content writer found this is so adorable, and then this study made a way in several scientific magazines, in the Nature, uh, uh, New York Times, and National Geographic. So about the mosquitoes, now I would like to remind you that female Aedes aegypti mosquito, they mate only once in lifetime. So one of the researchers from Boschel lab, Leah, actually came to us and she wanted to characterize uh, the terminal part or the terminal part of the mosquito. So what we did, and she also wanted to compare the virgin mosquito and the mated mosquito just to see why they mate once. Is there any structural changes or morphological changes happen during the mating after the mating process so that they can't do this, or is there any correlation between them? So we did the electron microscopy after a conventional fixation, and this is the terminal part. So this is the bursa, and these are the plates, vaginal, and these are the vaginal lips, and this is the vaginal opening here. And when we compared the virgin uh, genital organs with the mated ones, we couldn't see any difference. So sometimes, this is like the results are there is no difference. So the science is like a bad date. It's like a lot of effort and no payoff. So okay, so now I will introduce you the problem we faced for the SEM imaging, what we did for these two labs. So this is the so another researcher from Corona lab, Daniel Hezazi Pastor, came to us, and he was interested in characterizing the different sensilla present in the head region of the uh, larva of clonal radar ants. So we fixed the sample using conventional microscopy, scanning electron microscopy method. We did critical point drying and everything, and this is the outcome of the uh, process. And the image looks collapsed, means larva got collapsed during the process, chemical processing, during the chemical processing of the larvae. So we were not happy actually. Though we could see most of these sensilla present in here, we could see them, but we were not very happy with the results, the outcome. So we had to think about like what to improve the to get the image in a near native state. And besides that, uh, Leah from Bosch Lab, she was also interested in see how the male and female genitalia interact during the mating process. So there is another. So this is uh, getting SEM images of the Aegypti during mating is like two-step process. First of all, it's very hard to get the mosquito in the mating phase because the mating ends only for 14 seconds. So that's a too little time, and it seems really next to impossible to capture these male and female in the act. So Amalia and I had to brainstorm, and we thought, why not to freeze them if we can capture them in the air? So let's see. So and we thought that okay, if we could do that, and we have the cryo electron scanning electron microscopy, we can improve our results for these two things. So for before going to the cryo-electron microscopy, I thought let's do some systematic approach to improve the ant larvae images. So what I did, I tweaked some of the things. Maybe I increased the. So I thought like, okay, this collapse structure, maybe dehydration is the problem. So I increased the time of the dehydration. I doubled the time of the dehydration. Uh, it did not enhance the quality of the images. And then what I did a longer critical point drying, which is the longer status protocol present in our autosymmetry machine, which takes around 6 times more than the normal method. It also did not increase any, so improve the method. So we had to brainstorm. And for the mosquito, we thought, why not to freeze them if we can capture them in the air? So we talked to Leah. So she is Leah, and then we thought, okay, if Leah, you can capture these mosquitoes, you can we can put them in the liquid nitrogen directly. So what she did, so this is the lower chamber where there are male mosquitoes, and there are few female mosquitoes on the upper chamber. And she took some of the male mosquito and put in the female chamber, and they were like uh, then she found couple of mosquitoes in the act, and she captured some of those couples. So we were talking, Amalia and I. So you assume this is me and this is Amalia, and we were talking about cryosim chamber and budget. So I think I told Amalia, I think I found a solution of our problem. So Amalia asked what it is, and I thought that we have to buy a cryosim chamber for our machine. And she asked about the cost, and I told $180,000. And she told, sorry, Anurag, budget is tight, and I wish I could order that. Look at another solution. So we found that Jol actually have a Pursman cryoscium. So it is nothing as it's a cryo stuff which is made up of brass, and it retains the coldness for a long time. And for sticking the solution at the lower temperature, we had to use a rice glue. So this looked like simple solution. So what is, and then next morning I told like, okay, Amalia, I found this alternative solution. So Amalia asked like, what? And I said, okay, maybe a poor man's cryo SEM block would be useful. And she told, what is the cost? And I was like, $150. It is like 1% of the cost. And she told, let's order it. So this is like a very simple process. What I do, this is the cryo stuff, and I put the aluminum stuff, pasted samples on top of it with the rice glue, and I immerse them directly into the liquid nitrogen and let it equilibrate. Once they cool down, I remove the assembly and place in the SEM of ours. We use Joule JSM IT500 HR scanning electron microscope. And the settings we use is the low vacuum mode, 3 Pascal, 20 kilovolts, and 50 probe current. You can change these settings also to improve the thing. So this is the photo I took inside the microscope, and you can see the assembly. After a while, I took the pictures, let the eye sublimate a bit. It takes around 3 to 4 minutes. It depends upon sample. And this is the random leaf samples. I plugged it from the Peggy Rockefeller Garden outside of our building and put it here. One is the abaxial and one is the adaxial surface of the leaf. And as you can see that the images are pretty nice. This is like without processing anything. I just plugged it in and put them in the liquid nitrogen and put them in the microscope, and these are the pictures. I mean, I could have improved these pictures, but this is just to show you how the Kurzman cryo-CM works. So for our samples, we were hoping that it would work. And this is the larva I processed, the whole body of larva. You can see it's very fragile and it's very hard for me to, even stick on the carbon tape for SEM analysis. But when I do the Kurzman cryo SEM and it improved the structure a lot, it is near native state of the larvae. So I wouldn't say this is a Kurzman cryo-SEM. This is a cost-effective cryo-SEM method. So for the mosquitoes, for the couples, actually, so this is like the genital engagement. So we could capture those couples. You remember the last video I showed, the Leah was capturing them, and these are the mosquitoes in act. And then we captured them and we put them under in the SEM and took under the condition what I've shown, and they look amazing. You can get tons of information. So this is the female and this is the male, and you can see this is the genitalia of the male mosquito, and this is the female mosquito. How they engage. These are the lips, and this is the like the uh, tip of the genitalia of female, and this is the gonostyle. And what they found, like gonostyle, they tickle somehow on the tip of the genitalia of female, and female elongates uh, uh, the lips actually in order to do the mating process complete. So this is how we could find out like how this happens with the help of our uh, cost-effective cryo-SEM method. Okay, so here, so this was the cryo-SEM method, and we also recently was, so Orly from the Corona lab was interested in studying the time-dependent development of the larval internal organs. So to optimize that, we had to do the temp fixation protocol for larvae of the clonal radar ants. But there were like challenges in this process because this is the embedded sample of the larvae. But you can see like there are some air bubbles in the larval gut, and these air bubbles they are very nasty and they hinder uh, they don't let the uh, solution pass through. And another problem is like thick cuticle also hinders penetrating the fixative and other chemicals in the body of larvae. So these were the challenges, and there were not many protocols available. So we only could find protocols related to some other very different ant and the larvae of these ants. Uh, so we had to find out a solution for that. So we were again brainstorming, having coffee in our coffee room, and thinking what to improve. So Amalia found this method that let's do the chemical fixation but put some triton in there. So we try to put like 0.1% triton x100 to improve the method. And she also suggested let's use heptane because somebody used heptane for some of the protocols, heptane partitioning. So this we tried and they did whole process. And then what we did, this did not work. And then what we did, because I have a plant background, so I picked many leaf samples previously uh, in my previous workplace. So what I thought, like, let's do the vacuum infiltration. So beside having the Triton X100 and heptane in the fixative, I also applied vacuum. Thought that, okay, this air bubble will go out and the vacuum will suck in the solution inside the larvae. This also did not work. So next, we thought, like, let's do the high pressure freezing if this is successful. So Amalia found, like, okay, let's put sucrose and let's do it with sucrose and without sucrose, just to preserve the cellular osmolarity inside the larvae. So we did sucrose and without sucrose, we subjected to high pressure freezing and freeze substitution. This also kind of didn't work. And then in the last, we did a Triton X100 for 30 minutes. And then we washed off this fixative solution and put in the normal fixative without Triton X100. And the next day, we did the high pressure freezing and freeze substitution. And yeah, I mean, we also removed the head of the larvae in this case. So these are the outcomes. So we did the chemical fixation first time. You see nothing worked. There is nothing black in here. We fixed with the 0.1% Triton X100 or heptane. This was the result. So all these white thing, what you can see inside the larval body, it's the oil droplets or oil bodies, actually, the fat bodies. They should have been black if the solution was in. So we knew that it didn't work. Our protocol didn't work. When we did with the vacuum infiltration, no, we could only see a few, layers, a few parts actually of the larvae were black, but still we could see that the fat bodies were white. When we use sucrose with high pressure freezing and without sucrose, it clearly did not work. However, when we decapitated the larvae and used the 0.1% Triton X100 for 30 minutes and followed by high pressure freezing and freeze substitution, I mean, we could see the black color inside the larvae, and we were so happy. I think this worked. So we were happy, and we generated some of the pictures. So this is like this is unpublished data, so I'm not going to give any detail. Uh, but this is just to show you that this protocol worked, and these are some of the holes, the air bubbles we were talking about um, in the larval gut, and they were collapsing somehow. So this is the larvae, and this is one of the pictures we could generate, and we had many more pictures, and the protocol worked completely. So we are happy to show the data to only. And with that, I would like to thank everybody for patiently listening to me. And I'm here to answer all your questions. And if you have any questions, then please, I have the email address, so you can also send me an email, and Amalia or I will be happy to answer your questions. Thank you very much.
So anyone has any questions? And is there anything in the chat? Okay, Louis, could you just ask the question directly?
Sure, thank you. Very nice talk. I appreciate it. Two short questions, hopefully. Could you say what the glue was that you used to adhere them in the cryo conditions? And then secondarily, it would seem like when you transfer from the liquid nitrogen to the SEM, you're going to get a lot of frost condensation on the surface. How you eliminate that? Thank you.
Okay, so I will go back. So first question was the glue. So you have to use the rice glue. This is very important. So rice glue is very important. You have to use the rice glue in uh, because this glue has a property to remain adhered on the surface even in the cryo conditions. So we purchased Japanese rice glue from Amazon, but I guess you can buy any. You can take a picture of this and order it from Amazon. And the second thing, the second thing you were asking about ice sublimation. You just have to put in and wait. The ice will sublimate in the machine itself, in the SEM itself. So this is the picture I have taken just after putting the sample. There is a lot of ice, and as you can see here, it's still there is ice in some part of the tissue and in the aluminum stub, but some parts ice has been sublimated. So you have to find a way that it goes away automatically, but you have to find that area where the ice has sublimated already and start imaging them as soon as possible.
Han, you want to ask your question? You're muted, okay?
Yes.
And
Can you hear me?
yes.
Yep.
Oh, okay, thank you. Yeah, really nice talk. Thank you. I do have a question for the last part about the TM. I mean, I saw you using the acetone. Did you ever be interested change acetone? Like final, you're using the PO? I mean, I've been working with insects and plant samples. I think maybe the last transition solution I will be using the PO instead of acetone. I don't have any compare results, but I just feel like maybe the PO will be better try. I just say like discussion. Thank you.
Yes, I completely agree with you. We also use ethanol and propylene oxide for many of our TEM protocols. But in this case, nothing was working. I mean, acetone was not the problem because osmium has to penetrate beforehand. We couldn't see, in our case, you can see, we couldn't see any penetration of osmium here in this case. And either in this case, all the oil bodies were like white. So we need to find out a way where osmium penetrates and stains this inside, stain the tissue and the cells. So, but I agree. We could have also tried propylene oxide, but we already tried so many things, and this generates a lot of money for the person and the lab. So that's also we have to consider how many things we can try. But I completely agree with you that we could have tried propylene oxide instead of acetone. Yes.